electrophoresis is a process we can use to get something called a DNA profile. So a DNA profile is where we compare the differences in DNA between individuals based upon segments of DNA that we chop up with restriction enzymes, which will give us a unique pattern, and that's what we call the DNA profile, and that's what we can compare between individuals. So there's also applications of DNA profiling. A really big one, or well-renowned one, is in crime. So if we have a suspect and we're trying to work out who committed a murder using DNA samples, we can go through the process of electrophoresis to get DNA profiles and compare them and try and ping a crime on a particular suspect. So the first part to this is, if we imagine that we have two segments of, or two sort of samples of DNA. So this is going to be sample one. We'll go with our crime scene analogy because this is often one that people find easier to understand. So say we've got two segments of DNA. The green is some DNA from a crime scene that we've got, and the purple is a particular suspect. So someone is suspected of crime. We've got a DNA sample from them. Now, even though people have the same, so if one person or other have the same amount of chromosomes or have the same genes which program for similar stuff, their genes are obviously unique. So when we say their genes are unique or their genes are varied, the particular genes on the chromosomes are going to have somewhat different base sequences because of that variation. They're not all identical. Now, because of this difference in base sequences, if we use some restriction enzymes to try and cut up the DNA, the restriction enzymes, if we use two restriction enzymes on each of these sections of genetic material, these sections of DNA, they're most likely going to cut in different places because they're going to see their desired sequence that they like to cut at a different spot along this segment of DNA. So say we've got restriction enzyme 1, sees its desired sequence here on the first sample, and it sees its desired sequence here on the second sample, and then we have another restriction enzyme, and maybe we're going to see it cut like so, and then like so. You can notice there that we're going to have the DNA samples chopped up into different segments. Okay, so what we can then do is we can load these segments into wells in something called electrophoresis or gel electrophoresis in order to create a DNA profile, which is similar to an example of DNA profile we've got down here below. So how gel electrophoresis works is the light blue in this picture here is a gel. So we make this gel and then we allow it to solidify in a bit of a block. What will happen is this end will attach a negative electrode, and down the bottom here will be a positive electrode. So we run a charge through our gel. Now the reason why we do this is when we add, so let's say for example sample A here is our green segments, sample B are our purple segments. When we add our two different samples of DNA in the chunks that have been cut up, or the pieces that have been cut up with our restriction enzymes, when we add them into wells up the top here, so we've got, these are our wells along the top there, because DNA has a slight negative charge, the DNA is going to want to move in this direction from the negative electrode down towards a positive electrode. The smaller the section of DNA, the smaller, the shorter amount in base pairs the DNA is, the faster and further it's going to move down towards a positive electrode. Larger DNA segments are going to move not as far. So you can see that along the left-hand side here. 1,000 base pairs is obviously bigger than 100 base pairs, so 1,000 base pair chunks of DNA aren't going to move as far. Along left here we've got our marker, sometimes this is called a ladder, so we can add known segments of DNA into one of the wells, and this will spread out a nice kind of guide for size of DNA, and then we can put our sample chunks of DNA into another well and allow them to move down for a certain amount of time. Now we'll allow this to happen for a period of time until we get these segments of DNA spreading out, and then we can take something called an electrophoregram, which is kind of a snapshot of the movement of the samples that we've got. and this will give us an idea of our DNA profile. So what we can tell here then is if we add samples, so say we've got 3 samples here, sample A, sample B, sample C, what we can do is we can compare the similarities in DNA between different people's samples. So if we were to run I'll do a very quick little example for the side here. So say we were going to run some samples down a gel like so. If we had a result like this, I'll show you how we can kind of use it to interpret whether someone might have committed a crime or not. So if our green samples here are say crime scene DNA, so this is DNA retrieved from the crime scene, and we have retrieved DNA, I've got a DNA sample from suspect 1 and from suspect 2, like so. Now what we can tell about the crime scene DNA and the DNA we've got from one of our suspects, suspect 2, the electrophoregram we've got, the profile, is exactly the same. So we've seen the same movement of DNA chunks throughout the or DNA segments throughout the gel, which means that back in our restriction enzyme cutting phase, the crime scene DNA and the suspect 2's DNA have been chopped up in the same position, which would lead us to believe that they have the same sequence. Suspect 1, we see something different. So from this, we should be able to determine that both suspect 2 and the crime scene DNA came from the same person because they've been chopped in the same location due to having the same base sequence. So we've got this similar pattern in our DNA profile from gel electrophoresis. So that's a guide to how gel electrophoresis can be used in crime, along with a number of different applications, because of the way we can cut DNA with restriction enzymes. The variation will cut them into different size segments that will move differently through a gel to give us a DNA profile.